For over a century, some of the greatest minds in neurology spent their lives staring at the brains of schizophrenia patients through microscopes, and, well, they found absolutely nothing.
Nothing at all.
I mean, I want you to really think about that. These were brilliant pioneers, right? They had successfully mapped out the physical wreckage of conditions like Alzheimer's disease, Huntington's chorea, the late stages of syphilis.
Spot on, yeah.
They could take a slice of brain tissue, put it on a slide, and point directly to the plaque or the tangle that was basically destroying the patient's mind. But when they applied those exact same techniques to schizophrenia, the slides were clean.
Which is just staggering when you think about it.
Frustratingly normal tissue. It became such a notorious dead end that the eminent neurologist Fred Plum famously declared, "Schizophrenia is a graveyard of neuropathologists."
It is a brilliantly chilling quote, isn't it?
It really is.
And it captures an entire era of profound scientific frustration. It was basically the ultimate career ender. You could spend decades of your life peering into microscopes, hunting for the biological smoking gun of schizophrenia, and just retire with empty hands.
Which perfectly brings us to the core mission of our deep dive today. We are exploring the structural brain imaging of schizophrenia, and we're using Kaplan and Sadok's comprehensive textbook of psychiatry as our foundation.
Right.
Because ultimately, psychiatry is the study of how the brain, the mind, and human experience interact, and how that profoundly complex process can, well, go wrong.
Exactly.
But to understand how far we've come with modern imaging, we really have to understand the sheer scale of that early failure. Emil Kraepelin and Eugen Bleuler, the titans who first formulated the concepts of dementia praecox and the schizophrenias back in the late 19th and early 20th centuries, they were absolutely convinced this was a biological disease.
Oh, absolutely. They weren't looking for a purely psychological phenomenon at all.
Right.
They were watching their colleagues make massive inroads into understanding physical brain pathologies. I mean, Alois Alzheimer's is the perfect example here.
Oh, the Alzheimer's disease chap.
Yes. But what is often forgotten is that Alzheimer's actually tried to map the neuropathology of schizophrenia first.
Wait, really? He tried and just gave up?
He essentially abandoned it, yeah. He investigated the postmortem brain tissue of patients with schizophrenia, applying those silver staining techniques that were revolutionary at the time. But the tools were just, they were too crude.
And I suppose their expectations were probably wrong, too.
Entirely wrong. They were hunting for something large and dramatic, a gaping lesion, a massive crater of dead tissue. And when they didn't find it, the postmortem studies just yielded messy, conflicting results.
I can imagine.
One researcher would claim they found a subtle anomaly in one brain layer, and then the next researcher couldn't replicate it at all. This led to a massive waning of interest. For decades, a huge portion of the psychiatric community began to wonder if schizophrenia was purely a functional psychological disorder, with absolutely no physical footprint in the brain.
But the footprint was there. It was just hiding in plain sight, waiting for the right kind of light. And this brings us to a metaphor I really want to carry with us through this entire deep dive.
Okay, let's hear it.
I want you, the listener, to imagine stepping into a vast cathedral-like radiological observatory. We aren't here to look out at the stars, though. We are here to look inward at the living human brain.
I like that.
The marvel of this modern observatory is that we can examine the architecture of thought without ever lifting a scalpel. And in this space, we aren't looking for those massive craters that Kraepelin and Alzheimer's expected. We are looking for whispers.
Whispers, right.
Subtle, delicate shifts in the architecture. The gray matter, the white matter, the fluid-filled ventricles, and those incredibly intricate microscopic pathways that bind them all together.
And, that observatory was locked tight until about 1976. That was the watershed moment.
What happened in 76?
A researcher named Eve Johnstone and her colleagues used early computed tomography CT scanning. Now, by today's standards, those images are incredibly primitive, but at the time, it was an absolute revolution.
Because it was the living brain.
Exactly. For the very first time in human history, they could look inside the skull of a living patient experiencing schizophrenia.
Let's contextualize what a CT scan actually is for a moment, just so we are all on the same page. It's essentially a series of X-ray images taken from different angles, and a computer processes them to create cross-sectional slices of the bones, blood vessels, and soft tissues.
Right. It relies on tissue density.
Yeah.
And because it relies on density, it's very good at showing the difference between bone, brain tissue, and fluid. So Johnstone's team observed that the lateral ventricles, Those are the interconnected, fluid-filled cavities deep within the brain that produce and circulate cerebrospinal fluid. They were significantly enlarged in individuals with schizophrenia compared to healthy controls.
It's sort of like having a blurry black and white satellite photo of a landscape. You can't see the individual trees, but you can see a river.
Yes, precisely.
And Johnstone's team looked at that photo and said, right, that river is definitively wider than it should be. It was a single finding, but it was a seismic shift. It proved in vivo, in a living, breathing patient, that there was a structural physical difference. The graveyard wasn't empty after all.
It completely reopened the biological investigation into the illness.
Well,
But the true quantum leap, the thing that changes everything, happened in the late 70s and 80s when Peter Mansfield and Paul Lauterbur developed magnetic resonance imaging. MRI changed everything. We moved from that blurry satellite photo to a high-definition, three-dimensional Google Earth view of the brain. We could suddenly zoom in and see the wear and tear on the individual streets.
I think we need to unpack how MRI actually achieves that, because it's not just a better camera, is it? It's fundamentally different physics, which dictates what we can actually see.
It is.
Unlike a CT scan, which blasts you with ionizing radiation X-rays, an MRI uses incredibly powerful magnetic fields and radio waves.
It's a brilliant piece of physics, actually. The human body is mostly water, which means it's full of hydrogen protons. Normally, these protons are spinning on their axis in entirely random directions.
Just floating about.
Right. But when you slide a patient into an MRI scanner, you are placing them inside a massive superconducting magnet. That magnetic field forces all those tiny hydrogen protons in the brain to align themselves, kind of like microscopic compass needles, all pointing north.
And then comes the radio frequency pulse, right?
Exactly. The machine fires a specific frequency of radio waves at the brain. This pulse knocks the protons out of their alignment. When the radio pulse is turned off, the protons gradually realign with the magnetic field.
Like springs bouncing back?
Yes, and as they do, they release energy, which the scanner's coils detect. But here is the genius of MRI protons in different types of tissue, like gray matter, white matter, cerebrospinal fluid. They all realign at slightly different speeds.
Oh, I see.
By measuring those relaxation times, the computer builds an incredibly detailed, high-contrast map of the brain's soft tissue.
And crucially, because there is no ionizing radiation, there is no known adverse biological effect. You can scan someone safely multiple times over their entire lifespan.
Which is huge for research.
Right. You can scan children, you can scan adolescents who are at risk. It basically opened the door to longitudinal studies watching the brain physically change across time.
Which really defines our mission for this deep dive. Because of MRI, we are no longer asking if schizophrenia is a brain disease. We crossed that bridge decades ago.
Full stop.
Exactly. The field is now focused entirely on asking, what sort of disease is it? When does it start? How does it progress over decades?
Are we looking at a neurodevelopmental issue where the brain is built incorrectly? Or a neurodegenerative one where it breaks down?
Or perhaps both. So today, we're going to trace the illness chronologically from the chronic stages back to the first episode, and even earlier into high-risk populations, using structural and diffusion MRI as our guides.
Let's begin with the baseline. When we look at those high-definition images, what are the core differences we actually see in the gray matter? The gray matter being the processing centers of the brain, the densely packed neuron cell bodies, dendrites, and synapses.
The cities, if we stick to the map metaphor.
Exactly, the cities where the actual computational work of the brain occurs.
So to understand the baseline, we have to look at how the methodology of neuroimaging has evolved. In the 90s and early 2000s, it was quite common to see a published study featuring, say, 20 patients and 20 healthy controls.
Which sounds like a decent sample for an intensive scan.
It does, but the problem is that schizophrenia is extraordinarily heterogeneous. It presents differently in almost everyone. A slight structural difference you find in a group of 20 patients might just be statistical noise, or a quirk of that specific local sample.
It's the equivalent of trying to figure out the average height of a human being by measuring 20 people in a single pub in London.
Right.
You might get a completely skewed answer if a local basketball team happens to be having a pint.
That's a perfect analogy. So to find the true signal in all that noise, the field had to move to big data. And that is where the ENIGMA consortium comes in.
ENIGMA, what does that stand for?
It stands for Enhancing Neuroimaging Genetics Through Meta-Analysis. It is a massive global collaborative effort. Instead of a single lab doing a single scan, Inajima pools raw MRI data from thousands of individuals across dozens of independent research sites around the entire world.
I can't even imagine the logistical nightmare of that. You've got different clinics, different patient populations, and most importantly, different MRI machines.
Oh, it's incredibly difficult.
A 1.5 Tesla scanner in Tokyo is surely going to produce a slightly different image than a 3 Tesla scanner in London.
The data harmonization required to fix that is a monumental statistical achievement. They have to use complex algorithms to strip away the scanner-specific biases so they can actually compare apples to apples.
But when they do...
The statistical power is unprecedented. In 2016, Theo van Erp and his colleagues published an Inajima study focusing entirely on subcortical structures, so the deep, evolutionarily older regions of the brain. They analyzed scans from over 2,000 individuals with schizophrenia and over 2,500 healthy controls.
And what did the sheer weight of that data reveal about the deep brain?
It revealed robust, undeniable volume reductions in several key structures. Specifically, they found smaller volumes in the hippocampus, the amygdala, the thalamus, and the nucleus accumbens.
Wow.
All alongside an overall smaller intracranial volume and confirming that 1976 CT scan, larger lateral ventricles.
Let's unpack those structures for a moment, because we shouldn't just list them. We need to understand what a reduction implies clinically for you as a practitioner.
Absolutely.
The hippocampus is central to memory formation and indexing, turning short-term experiences into long-term memories. The amygdala is the emotional processing center, particularly attuned to salience and fear detection.
Right.
The thalamus is the grand relay station of the brain, routing sensory information up to the cortex. And the nucleus accumbens is the heart of the brain's reward and motivation circuitry.
When you lay it out like that, it perfectly mirrors the clinical presentation of schizophrenia.
It really does.
You've got memory deficits, abnormal emotional processing and heightened paranoia, sensory hallucinations from the relay station misfiring, and the profound lack of motivation. The negative symptoms like anhedonia and avolition. The structural deficits map directly onto the clinical reality.
So that's the deep brain. What about the outer layer, the cortex, the most highly evolved, complex part of the human brain?
Well, 2 years later, in 2018, Van Erp led another massive Inajima analysis, this time focusing on cortical thickness and surface area, pooling over 4,400 patients and 5,000 controls.
Massive.
Truly massive. And they found a global, widespread reduction in cortical thickness. But it wasn't just a uniform shrinkage.
Oh.
The thinning was highly regional, particularly prominent in the ventrolateral prefrontal cortex, the insula, and the anterior cingulate cortex.
Again, mapping onto the symptoms.
Exactly.
The prefrontal cortex manages executive functioning, working memory, and complex decision-making areas profoundly impacted in schizophrenia. The insula is heavily involved in interoception, our sense of our own internal physiological state, and self-awareness, which might relate to insight.
Yes, a lack of insight is huge in these patients.
And the anterior cingulate is crucial for cognitive control and error detection. It's a very logical map of decay. But, and correct me if I'm wrong, I recall from the chapter that it wasn't entirely a story of tissue loss. There were areas where the cortex was actually thicker, weren't there?
Yes, you're right. And this is where the complexity of the illness really shows itself. They observed anomalous areas of thicker cortex in the primary motor, somatosensory and parietal areas.
Thicker? Why would it be thicker?
This isn't entirely understood yet, but it's a crucial finding because it proves we are not just looking at a brain that is uniformly withering away. We're looking at a brain undergoing complex reorganization. It is miswired, aberrantly constructed in some regions, and lacking in others.
Okay, I want to stop right here because I think a lot of listeners might be having the exact same thought I am.
Go on.
We have this unbelievable, harmonized data from nearly 10,000 people. We know exactly which deep structures are smaller. We have a highly detailed topological map showing exactly where the cortex is thinning and where it is thickening.
Right.
So why in the year 2026 can a psychiatrist not just order an MRI to diagnose schizophrenia? A patient walks into the clinic exhibiting paranoia and hallucinations. Why not just put them in the scanner, look for a small hippocampus and a thin anterior cingulate, and say, right, there's your diagnosis?
It is the most common question asked by medical students, and the answer lies in understanding statistical effect sizes.
Effect sizes, okay.
In research, an effect size, often measured as Cohen's d, tells you the magnitude of the difference between two groups. In the Inajima studies, the single largest effect size they found was the reduction in the volume of the hippocampus, which had a Cohen's d of -0.46.
So an effect size of -0.46. Let's translate that out of statistical jargon for you listening.
Right, so a Cohen's d of zero point 0.2 is considered a small effect, 0.5 is medium, and 0.8 is large. So negative 0.46 is a medium effect. What this means in practice is that if you take 1,000 people with schizophrenia and 1,000 healthy controls, the average hippocampal volume of the schizophrenia group will be definitively, statistically significantly smaller.
But,
and this is the critical caveat, the bell curves of those two populations overlap massively.
right, it's a population-level truth, but not an individual diagnostic stamp. It's like saying that, on average, men are taller than women. Statistically, that is a robust, replicable fact with a very large effect size. But if a patient walks into your clinic and you measure them at 5'9", you cannot use that single piece of data to diagnose their gender.
No, of course not.
There are millions of women who are 5'9", and millions of men who are 5'9".
That is an excellent analogy. There are millions of perfectly healthy, neurotypical individuals walking around right now who happen to have a naturally smaller hippocampus or a thinner prefrontal cortex than a patient with severe schizophrenia.
So the overlap is just too great.
Precisely. The group differences are profoundly important for understanding the underlying biology and pathology of the disorder across humanity. But they are far too subtle, and there is far too much overlap with healthy human variation to diagnose the individual sitting in front of you.
Which naturally forces us to ask the next logical question. If these structural differences exist, when do they actually happen? Is a patient born with a smaller hippocampus?
Good question.
Does it fail to grow during adolescence? Or does it shrink after the illness begins? We need to become time travelers.
We do.
Yeah.
And to understand the trajectory, we should start at the end and work our way backwards. Let's examine chronic schizophrenia.
For a very long time, the prevailing wisdom in the graveyard of neuropathologists was that if there were brain abnormalities, they were static.
Yes.
The damage occurred early, perhaps in the womb or during birth, and then just sat there unchanging for the rest of the patient's life.
That was the neurodevelopmental hypothesis in its purest, most rigid form. But modern longitudinal MRI has fundamentally rewritten that narrative. We now have compelling evidence for a process of accelerated aging in the brain.
Accelerated aging.
Yes. A landmark study published by Cropley and colleagues in 2017 provides some of the clearest evidence. They didn't just look at a snapshot. They looked at the trajectory of 326 subjects with chronic schizophrenia.
And what did that trajectory reveal about the rate of change?
It revealed that gray matter loss doesn't just happen once. It is ongoing, but at a highly variable speed depending on the patient's age. They found that between the ages of roughly 20 and 45, patients with schizophrenia exhibited a 30% to 60% faster rate of gray matter volume loss compared to age-matched healthy controls.
A 30 to 60% faster rate of loss. Let's really think about that.
It's massive.
We all lose a bit of brain volume as we age. It's a natural part of human senescence. But imagine a biological clock ticking inside the architecture of the brain. And for 2 decades of early adulthood, that clock is running wildly fast.
Right.
The brain is weathering a lifetime's worth of aging, a lifetime's worth of cellular wear and tear and synaptic pruning, compressed into the span of just 20 or 25 years.
It is a profound structural weathering. But the Cropley study found something else, something actually quite unexpected.
Yeah.
It doesn't stay that fast forever.
It doesn't.
No. After the age of 50, the rate of gray matter loss in the schizophrenia cohort actually began to slow down. In older age, their trajectory of tissue loss paralleled the normal aging process of the healthy controls.
That's fascinating. So the accelerated damage is highly concentrated. It's not a relentless, lifelong decay like Alzheimer's.
Right.
It's a massive biological storm that rages through early adulthood and then eventually blows itself out, leaving the brain altered but relatively stable in its later decline.
Precisely.
Yeah.
Which tells us that the early phases of the illness are structurally the most dangerous. So let's rewind the clock further. Let's move back from the chronic phase, back into the patient's 20s, to the very first episode of psychosis.
The moment the illness truly breaks the surface.
Exactly.
This is where we have to discuss the Iowa longitudinal study led by the legendary Nancy Andreessen in 2011. This is one of those studies that just leaves you in awe of the sheer stamina of the researchers.
Well, without a doubt.
They prospectively followed 543 first-episode psychosis patients. And they didn't just scan them once or twice. They followed them for up to 18 years, collecting nearly 1,000 MRI scans.
It is a monumental piece of science. I mean, keeping an actively psychotic, struggling patient cohort engaged in a neuroimaging study for nearly 2 decades requires immense clinical empathy and logistical brilliance.
Truly.
And they wanted to answer a specific question. Are brain changes progressive right after the onset of the illness?
And after 18 years of tracking, what did the scanners show?
The data revealed that the transition to psychosis is a period of intense, almost violent biological turbulence. They found that 34% of the patients showed a 0.5% yearly brain volume reduction, and 14% showed a severe 1% yearly reduction.
A full percent every year?
Yes. But the most vital insight, the thing every clinician must keep in mind, is the timing. The most dramatic, rapid tissue loss, particularly in the frontal gray and white matter, did not occur evenly over the 18 years. It occurred predominantly in the first 2 years after the illness onset.
The first 2 years. So the storm is at its absolute most destructive exactly when the clinical symptoms are exploding for the first time.
Exactly.
The brain is undergoing a macroscopic structural crisis at the exact same moment the mind is undergoing a psychological crisis. The hallucinations, the delusions, the disorganization, they are mirrored by a rapid collapse of frontal tissue.
Yes, and as the brain tissue shrinks, the lateral ventricles, those fluid-filled spaces Eve Johnstone saw back in 1976, they passively expand to fill the void.
Just taking up the slack.
Right. And this isn't just an interesting biological quirk. It has deep, profound clinical meaning. The Iowa study found a direct correlation between the structural integrity of the brain and the clinical outcome of the patient.
In what way?
Individuals who had the smallest increases in cerebrospinal fluid, meaning they lost the least amount of cortical tissue during that critical 2-year window, had the longest subsequent periods of clinical remission.
Wow.
The severity of the biological storm at onset literally dictates the trajectory of the rest of their lives.
Which forces us to ask the ultimate question. Why? Why is the brain suddenly losing tissue at this terrifying rate during this specific early phase? It's not just disappearing into the ether. Something biological is destroying it or clearing it away. What is the mechanism?
We are stepping onto the absolute frontier of psychiatric research here. The graveyard of neuropathologists is full of failed theories about mechanisms. But right now, one of the most compelling and heavily researched frameworks is the immune dysregulation hypothesis.
Immune dysregulation.
Yes. We are accumulating evidence that the peripheral immune system and the central immune system inside the brain are deeply involved in this rapid tissue loss.
Okay, let's unpack this. How do we link the immune system to a shrinking prefrontal cortex?
Let's look at a very elegant study by Nunez and co-authors. They examined 137 individuals experiencing their first episode of psychosis, but they didn't just put them in an MRI scanner. They also drew their blood. They combined the radiological observatory with the pathology lab.
I like that approach. Looking at the entire systemic picture, what were they looking for in the blood?
They were looking at basic inflammatory markers, specifically neutrophils. Neutrophils are a type of white blood cell. They are the innate immune system's first responders to infection or inflammation.
Right.
The researchers found that compared to healthy controls, the first episode patients had significantly higher neutrophil counts and conversely lower red blood cell counts.
So their peripheral immune system was activated. They were in a state of systemic inflammation.
Yes, but the breakthrough was mathematically linking the blood to the brain. When they cross-referenced the blood panels with the MRI scans, they found a direct correlation in the early psychosis group.
Which was?
Higher neutrophil counts in the blood were directly associated with smaller gray matter volumes, particularly in the right anterior insula and the temporal pole.
Wait, that is quite a profound leap, isn't it? The more inflamed the peripheral immune system was, the more white blood cells coursing through their veins, the less gray matter they had in their brain.
Exactly. And to tie it back to the clinical reality, those higher neutrophil counts didn't just correlate with smaller brains, they correlated with higher PANAS scores.
The positive and negative syndrome scale.
Right. Meaning the patients with the most inflammation and the least gray matter were also suffering from the most severe positive symptoms, like acute hallucinations and entrenched delusions.
Now, I have to jump in here and push back a bit because there is a massive chicken or egg dilemma staring us in the face.
I thought you might bring that up.
I mean, having your first psychotic break, losing your grip on reality, experiencing terrifying hallucinations, perhaps being involuntarily hospitalized, has to be one of the most stressful, traumatic events a human being can endure.
Oh, absolutely.
We know as a physiological fact that extreme psychological stress spikes cortisol. We know chronic stress activates the peripheral immune system. Is it not highly plausible that the sheer terror of the psychotic episode is causing the systemic inflammation rather than the immune system attacking the brain and causing the psychosis?
It's a brilliant question, and it is the exact dilemma that keeps neuroscientists up at night. You are entirely correct that stress induces inflammation. However, the emerging models suggest a more insidious central mechanism. We need to talk about microglia.
Microglia, the resident immune cells of the central nervous system.
Yes. The brain is separated from the peripheral immune system by the blood-brain barrier. It has its own dedicated immune force, the microglia. During normal brain development, especially in adolescence, microglia act as gardeners.
Gardeners?
Yeah, they prune away weak or unnecessary synapses to make the brain more efficient. The theory is that in schizophrenia, perhaps triggered by a combination of genetic vulnerability and environmental stress, these microglia go rogue.
They start over-pruning.
Exactly. They become overactivated and start aggressively over-pruning synapses, stripping away the neuropil, the dense network of dendrites and axonal branches.
So the gray matter loss we see on an MRI isn't necessarily whole neurons dying and disappearing. It's the connections between them being aggressively sheared away by the brain's own immune system.
That is the leading hypothesis. It's a reduction in neuropil, not widespread neuronal death. And this intense central neuroinflammation might then leak signals across the blood-brain barrier, triggering the peripheral immune response, the elevated neutrophils that Nunez measured. We don't have the definitive causal arrow yet, but the link between early tissue loss and immune activation is undeniable, and it opens up a revolutionary clinical avenue. What if we treated early first-episode psychosis, not just with dopamine-blocking antipsychotics, but with highly targeted central nervous system anti-inflammatories? Could we stop the biological storm and save the tissue?
That would fundamentally alter the trajectory of the disease. It's an incredibly hopeful avenue of research. But you mentioned genetic vulnerability triggering this response. We've known for over a century that schizophrenia runs in families.
Right.
The heritability is high. How are we connecting the genetic blueprints to the macroscopic tissue loss we see on an MRI?
This requires integrating two massively complex fields, genetics and neuroimaging. And a study by G and colleagues provides a fascinating blueprint for how to do it. They performed what is called a transcription neuroimaging association analysis.
Okay, let's break that down for the listener. What is that?
Over the past decade, genome-wide association studies, or GOE ways, have identified hundreds of tiny genetic variations across the human genome that increase the risk of schizophrenia. But knowing a gene variant exists doesn't tell you what it actually does in the brain.
Right, it's just a marker.
Exactly. So Gene and colleagues took 98 of these highly associated schizophrenia risk genes, and they mapped them onto the Allen Human Brain Atlas.
And the Allen Brain Atlas is basically a highly detailed, three-dimensional post-mortem map of where specific genes are actively expressed or turned on in the human brain.
Precisely. So they have a 3D map of where the risk genes are expressed. Then they took harmonized MRI data showing exactly where gray matter volume loss occurs in living patients. They overlaid the two maps to see if the regions of the brain that shrink the most are the same regions where the risk genes are most active.
And did they match up?
They found significant correlations across the board, but one specific gene stood out with a remarkably strong association, a gene called GRIA1.
Let's unpack GRIA1. What is its day job in a healthy brain?
GRIA1 is a gene intimately responsible for encoding a subunit of the AMPA receptor. Now, the AMPA receptor is an absolutely critical component of glutamatergic transmission.
Okay.
Glutamate is the primary excitatory neurotransmitter in the brain. When one neuron wants to send a rapid signal to another, it releases glutamate across the synapse, which binds to AMPA receptors, triggering an electrical charge in the receiving neuron.
And beyond just sending a signal, AMPA receptors are heavily involved in synaptic plasticity, aren't they? The ability of synapses to strengthen or weaken over time, which is the fundamental physical basis of how we learn, adapt, and form memories.
Exactly. Long-term potentiation, the strengthening of synapses, relies heavily on the density and function of AMPA receptors. So what G and colleagues found was that higher levels of GRA1 expression in specific brain regions were associated with a greater degree of gray matter volume loss in those exact same regions in patients with schizophrenia.
That is an incredibly elegant, if devastating, piece of biology. From a tiny genetic variant on a chromosome to a faulty AMPA receptor, to disrupted glutamate signaling and synaptic plasticity, resulting in the brain aggressively pruning its own architecture, which we finally see as macroscopic tissue loss on an MRI scan.
Yes.
The hardware of the brain is shrinking because the microscopic software regulating the connections is faulty.
It connects the micro to the macro beautifully. It suggests the tissue loss isn't random, it is guided by the genetic blueprint.
So we've looked at the chronic illness, and we've looked at the intense biological storm of the first episode. But what about before the first episode? If the genetic vulnerability is there from birth, can we see the storm gathering before it makes landfall?
This is arguably the most clinically urgent area of psychiatric research today. The study of clinical high risk, or CHR, populations. We are talking about young people, typically older adolescents or young adults, who are experiencing what we call the prodrome.
The prodrome being the warning signs. They haven't had a full psychotic break. They aren't fully detached from reality, but they are experiencing subthreshold symptoms.
Exactly.
Perhaps they are hearing faint whispers that they know aren't real, or their thoughts are becoming slightly disorganized, or their social functioning and academic performance are suddenly dropping off.
Right. They're in a state of profound vulnerability. Now, we know epidemiologically that only a fraction of these CHR individuals will actually transition and convert to full-blown schizophrenia. It is usually less than a third.
And the rest?
Many will simply recover or transition into a different mood disorder. So to study them effectively, you need massive longitudinal consortia to capture enough people who eventually convert.
And one of the most prominent is the North American Prodrome Longitudinal Study, or NAPLS.
Yes. In 2015, Cannon and colleagues published a vital NAPLS study looking at 274 clinical high-risk cases. They scanned them at baseline and then tracked them over time. During the follow-up period, 35 of those individuals converted to full psychosis.
Okay, so this is the crucial moment. What did their MRI scan show at the very beginning, at baseline, before anyone had converted? Could you look at the scans and predict who was going to break?
At baseline, the observatory was quiet. There were absolutely no significant structural differences between the CHR individuals who would eventually convert, the CHR individuals who wouldn't convert, and healthy controls.
Wow. So even when they're experiencing prodromal symptoms, the macroscopic structure of the brain still looks completely intact.
But then they tracked the longitudinal changes. Over the next 12 months, as that subset of 35 individuals transitioned into full psychosis, their brains changed dramatically. The converters exhibited a significantly faster rate of gray matter loss, specifically in the right prefrontal cortex, and a significant expansion of the third ventricle compared to the non-converters and the healthy controls.
The structural collapse was happening in real time, exactly parallel to the psychological collapse. Now, I have to play devil's advocate here, because this is where the clinical reality collides with the research.
Go ahead.
If an adolescent is in a clinic, highly distressed, experiencing disorganized thoughts and attenuated hallucinations, a psychiatrist is very likely to prescribe an antipsychotic medication to try and alleviate that distress and prevent a full break.
That is the standard of care in many situations, yes.
But we know that antipsychotics are powerful, systemic chemicals that alter dopamine pathways globally in the brain. Is it not entirely possible that the rapid thinning of the prefrontal cortex we are seeing on the MRI isn't the disease at all, but rather a neurotoxic side effect of the heavy medications we are giving these young people?
It is an absolutely vital question, and it has been the source of fierce debate. But the NAPLS study anticipated this and addressed it directly. They looked specifically at the subset of converters who had zero exposure to any antipsychotic medications during the period between their baseline and follow-up scans.
And what did the unmedicated brains show?
The unmedicated converters actually showed a greater reduction in prefrontal cortical thickness than the non-converters. The presence of medication did not explain the thinning. In fact, the thinning was most pronounced in those who transitioned to psychosis without any pharmacological intervention.
So the medication isn't causing the initial collapse. The thinning is the fundamental pathology of the illness onset itself.
Exactly. It validates that the transition to psychosis is an inherently biologically destructive event for the cortex. But what if we look even earlier? What about individuals who aren't showing any symptoms at all, no prodrome, no whispers, but simply have a strong family history, the genetic high-risk group?
Right. Say the healthy 18-year-old sibling of someone with severe schizophrenia. They share roughly 50% of their DNA, so they carry a heavy genetic load. But they haven't suffered the neurotoxic effects of a psychotic episode, and they certainly aren't taking antipsychotics. They are a pure window into vulnerability.
Precisely. A 2014 review by Bois and colleagues looked at exactly these familial high-risk populations. And they found that even without being ill, these unaffected family members show decreased gray matter volume in prefrontal and temporal areas compared to healthy controls without a family history.
So the genetic blueprint leaves a physical fingerprint on the brain's architecture, even if the building itself never collapses.
The vulnerability is physically manifest, but the longitudinal data is where it gets truly fascinating. Over time, healthy adolescents without genetic risk and even high-risk individuals who never convert to illness exhibit a healthy, normal trajectory of cortical thickening and maturation as they move through their teenage years into adulthood.
The brain is building its adult scaffolding.
Yes, but the high-risk individuals who do eventually go on to convert show that characteristic anomalous reduction in cortical thickness. They physically diverge from the healthy developmental trajectory under the stress of maturation.
It's a brilliant, if tragic, metaphor. The genetic blueprint dictates the construction of a fragile scaffolding. For many siblings, despite the genetic risk, the scaffolding holds up through the turbulent weather of adolescence. But for those who convert, the scaffolding simply cannot bear the structural load, and it begins to give way.
That's a very apt way to conceptualize it. And speaking of scaffolding and structural integrity, we need to shift our focus entirely.
Okay, where to?
Up until now, we spent this entire deep dive talking about gray matter. We've talked about the cities of the brain, the processing centers shrinking and thinning. But a brain isn't just a collection of isolated cities, it is a network. How does information actually travel between a thinning prefrontal cortex and a shrinking hippocampus?
If the cities are in decline, what is happening to the highways connecting them? Which brings us to the disconnectivity hypothesis and the white matter. Now, looking at gray matter volume is relatively straightforward with a standard structural MRI, but how on earth do we image white matter in a living brain?
We use a remarkably sophisticated technique called diffusion MRI, or dMRI. To understand it, we have to understand what white matter actually is. Gray matter is the cell bodies. White matter is the billions of axons, the long, wire-like projections that extend from the cell bodies to connect with other neurons. And crucially, these axons are wrapped in myelin.
Myelin being the fatty, lipid-rich insulating sheath produced by oligodendrocytes.
Exactly. I like your metaphor of cities and highways. But perhaps a better one for white matter is a massive bundle of transatlantic fiber optic cables. The myelin is the thick rubber insulation around the cable. It prevents the electrical signal from leaking out, ensuring it travels incredibly fast and arrives intact at its destination.
So how does an MRI see a fiber optic cable?
It doesn't see the cable directly. It sees the water moving around it. Diffusion MRI measures the microscopic random thermal movement, the diffusion of water molecules in the brain tissue.
Okay, let's break down the physics of that.
Well, if you put a drop of ink in a glass of water, the molecules diffuse randomly in all directions, creating a perfect sphere. That is called isotropic diffusion.
Right.
But inside the brain's white matter, water molecules are trapped inside and around these long tube-like axons, which are tightly bundled and wrapped in fat. The water can't move side to side very easily because the myelin insulation blocks it. It can really only flow back and forth along the longitudinal length of the axon. This restricted, highly directional movement is called anisotropic diffusion.
So we measure how restricted the water is.
Yes, using a metric called fractional anisotropy, or FA.
Let me see if I have this right. A high FA value means the water is flowing smoothly and strictly in one direction along the axis, which implies that the fiber optic cable is totally intact, the myelin insulation is thick, the axons are tightly packed, and the network is healthy.
Correct.
Conversely, a low FA value means the water is diffusing more randomly, leaking sideways, which implies the cable is degraded, the insulation is stripped away, and the structural integrity of the network is failing.
You've got it perfectly. And when researchers applied dMRI to schizophrenia, it birthed the disconnectivity hypothesis. The illness arises not just because the prefrontal cortex is thin, but because the cables connecting the prefrontal cortex to the rest of the brain are degraded. The signals are corrupted, delayed, or lost entirely, leading to disorganized thoughts and hallucinations.
Did the Ian and Jemay consortium look at white matter?
They did. In 2018, they conducted a mega-analysis of dMRI scans from over 4,000 individuals, and they found widespread, significant reductions in fractional anisotropy in patients with schizophrenia.
Were there specific cables that were more damaged than others?
Yes. The deficits were particularly severe in the anterior corona radiata, which connects the cortex to the brainstem, and the corpus callosum.
The corpus callosum being the absolute largest superhighway in the brain, a massive bridge of hundreds of millions of axons connecting the left and right hemispheres.
Exactly.
If that bridge is degraded, the two halves of the brain literally cannot communicate effectively. But white matter isn't fully built when we are born, is it? It takes a very long time to lay down all that myelin. It continues maturing well into our 20s. How does schizophrenia affect that construction schedule?
This is where we see the true power of data harmonization. A researcher named Seton Kara-Umack led a phenomenal study. They took DMRI data from multiple different scanners and sites. As we discussed earlier, pooling DMRI data is notoriously difficult because different magnetic gradients produce different FA values.
Right, the different machines issue.
But they applied complex machine learning algorithms to remove the scanner-specific biases, allowing them to map white matter trajectories across the entire lifespan, from age 14 all the way to 65.
A unified lifespan map of the brain's network. What did the timeline reveal?
It revealed a tragedy of timing. In healthy controls, white matter FA, which we use as a proxy for the maturity and integrity of the myelin, peaks at around age 33. The network is fully built, heavily insulated, and optimized by our early 30s. But in patients with schizophrenia, FA reached its absolute peak at age 27.
Wait, it peaks 5 years earlier.
Yes. The brain of someone with schizophrenia seemingly rushes its own maturation. It finishes the construction schedule 5 years prematurely, peaking at a lower overall level of integrity, and then immediately begins a long, premature trajectory of decline.
It's a disease of a brain that rushed its own construction, built a faulty network, and then immediately started falling apart. Did they find different patterns of decay in different regions?
They found 3 distinct temporal patterns. First, early neurodevelopmental abnormalities in the limbic tracts, like the cingulate. These deficits are present early and remain relatively stable. Second, they found that abnormal rushed maturation in the association fibers, like the superior longitudinal fasciculus. And third, they observed a progressive, relentless deterioration, beginning right from the onset of the illness in the commissural tracts, primarily that massive bridge, the corpus callosum.
The architecture is just fundamentally out of sync with healthy human development. But let me bring up the inflammation angle again. We talked about neutrophils and microglia aggressively pruning gray matter. What about the white matter?
Good question.
If FA is low, it means water is moving abnormally. Can standard DMRI actually tell us if the transatlantic cables are physically broken, like the myelin is actively destroyed, or if the tissue is just inflamed and flooded with fluid?
This is the absolute cutting edge of diffusion imaging. You've hit on a major limitation of standard fractional anisotropy. Standard FA is a blunt instrument. It cannot reliably differentiate between a physical breakdown of myelin and the presence of neuroinflammation or edema swelling. But a new mathematical technique called free water imaging can.
Free water imaging, how does it do that?
It applies a bitensor model to deconstruct the diffusion signal into 2 separate compartments within the same voxel of tissue. It measures the restricted water trapped inside the myelin sheaths, which we call tissue FA, or FAT, and it measures the unrestricted free-floating water in the extracellular space outside the cells.
Okay, so tissue FA tells us about the physical health of the axon and myelin, and free water tells us if the space around the cable is flooded.
Exactly. In a landmark study by Lyle and colleagues, they applied this free water imaging technique to patients experiencing their first episode of psychosis.
What did they find when they split the signal?
When they just looked at standard FA, the scan showed the classic picture: widespread white matter damage across the brain. It looked like the network was already heavily degraded at onset, but when they split the signal, the narrative completely flipped.
Oh.
They found that there were widespread, highly significant increases in free water.
Meaning widespread neuroinflammation and edema. The brain was flooded.
Yes. But the actual reductions in tissue FA, the actual physical damage to the myelin sheaths and axonal integrity, were very small and highly localized.
That is a staggering revelation. It completely rewrites our understanding of the early illness. It means that at the onset of psychosis, the fiber optic cables aren't completely destroyed. They are just flooded with inflammatory fluid. And if they are flooded due to an immune response rather than permanently physically degraded...
I know exactly what you are thinking, and you are right. It implies a critical golden window for clinical intervention. It suggests that early psychosis involves significant and potentially reversible neuroinflammation rather than pure, irreversible neurodegeneration.
Which means we could treat it.
If a clinician can intervene early enough, perhaps with targeted anti-inflammatories, and drain that extracellular flooding, They might be able to protect the underlying myelin from the long-term progressive deterioration we see in chronic patients.
It offers profound hope, but it also transitions us perfectly to the clinical reality of how we are treating these patients right now, which brings us to the medication conundrum. We've established that antipsychotics don't cause the initial cortical thinning in high-risk patients, but what happens after a patient has been on these incredibly powerful dopamine antagonists for 10, 20, or 30 years?
It is the most difficult, ethical, and clinical tightrope a psychiatrist has to walk. And the neuroimaging evidence is incredibly complex and sometimes seemingly contradictory. Let's look at a vital study by Luwu and colleagues focusing on gray matter in chronic schizophrenia. They compared 3 groups: never treated chronic patients, patients treated long-term with a standard atypical antipsychotic like risperidone, and patients treated with clozapine.
Clozapine being the absolute gold standard, heavy-duty antipsychotic, usually reserved for the most severe treatment-resistant cases because of its serious side effect profile. What did the MRI scans show?
They found that while all the patients, regardless of medication, had significant gray matter loss compared to healthy controls, the treated patients showed more severe gray matter reductions than the never treated patients.
Wait, the medication exacerbated the tissue loss.
It appears so. And specifically, clozapine was linked to the most severe prefrontal gray matter reductions. Furthermore, this volume loss was negatively correlated with the dosage. The higher the cumulative dose of clozapine a patient had received over their lifetime, the greater the volume of cortical tissue they had lost.
That is deeply sobering. It's terrifying for a patient or family member to hear that the medication that is actively saving them from the torment of psychosis might be slowly accelerating the erosion of their cortical gray matter over the decades.
It is a harsh reality. But in psychiatry, we always have to look at the whole picture, the entire system. Let's pivot and look at the white matter. A different study by Xiao and colleagues used diffusion MRI to look at never-treated chronic patients versus treated patients.
And what did the white matter cable show?
They found the exact opposite effect. The never-treated patients showed a significantly faster age-related decline in their white matter FA compared to the treated patients.
So let me synthesize this, because this is the crux of the conundrum. The medications might exert a slow neurotoxic effect on the gray matter volume over time, accelerating cortical thinning. But simultaneously, they potentially protect or at least significantly slow the deterioration of the white matter integrity.
That is the current working hypothesis. It perfectly illustrates the impossible balancing act. You are intervening in a phenomenally complex, highly interconnected organ. Dampening dopamine and other neurotransmitter systems to halt a devastating psychotic break might have long-term structural costs to the cortical cities. But by stopping the psychosis and reducing the central inflammation, it might reduce the toxic downstream effects that degrade the white matter highways.
So it's a trade-off.
Clinicians have to weigh these long-term theoretical structural trade-offs against the immediate, devastating, and often life-threatening psychosocial risks of leaving acute psychosis untreated.
It's a choice between 2 incredibly difficult realities. But as we discuss all of this, something fundamental has been bothering me.
What's that?
We have spent the last hour talking about patients with schizophrenia as a monolithic group. We've talked about enigma averages and means and medium effect sizes. But if I am a patient sitting in a clinic, I do not care about the group average. I care about my brain. How do we move from these massive statistical analyses of 4,000 people to the individual human being?
That is the most important philosophical and technological pivot occurring in neuroimaging today. The limits of case-control group studies comparing the average of Group A to the average of Group B are becoming glaringly obvious. To understand why, we look at a brilliant study by LV and colleagues using a technique called normative modeling.
Okay, let's explain normative modeling. I imagine it like pediatric growth charts. When a parent takes a child to the doctor, the child's height and weight are plotted on a curve that was generated from millions of healthy children. The doctor doesn't compare the child to the average sick child. They compare them to the healthy normative range to see if they're deviating.
That is exactly what it is, but applied to the architecture of the brain. Kivi and colleagues built a massive healthy normative model for cortical thickness. Then, they took 322 individual patients with schizophrenia and plotted their specific individual MRI scans against that healthy growth chart.
And what happens when you look at the individual instead of the group?
When they ran a standard group comparison on those 322 patients, they found the classic result we discussed earlier. statistically significant reductions in cortical thickness in 60 out of 68 brain regions. The average patient looked very sick. But when they looked at the individuals using the normative model, they found something that shook the field. They found that up to 82% of individual schizophrenia patients fell entirely within the normative range of healthy brain thickness.
I need to stop you there because I want to make sure I'm hearing this correctly. You are telling me that despite everything we have discussed, the accelerated aging, the microglial pruning, the immune storms, the genetic vulnerability, if you take an individual patient with schizophrenia and look at their cortex, there is an 82% chance that their cortical thickness is statistically completely normal.
Yes. Only 10 to 15% of the individual patients had what they termed infranormal deviations, meaning statistically significant pathological thinning in the temporal or prefrontal cortices.
That is staggering. It completely shatters the idea that there is a single identifiable schizophrenia brain.
It does. It highlights the extreme radical heterogeneity of this disorder. Case control averages blur out the individual reality to find a weak mathematical signal. The reality is that the pathology in your brain might be severe thinning in the prefrontal cortex. But in the patient sitting next to you in the waiting room, their cortex might be perfectly intact, and their illness is entirely driven by degraded white matter connectivity.
And a third.
In a third patient, their networks might be fine, but their brain is flooded with immune-free water. If over 80% of individual scans look normal on a macro level, we absolutely cannot keep treating everyone with the exact same broad-spectrum medication.
So where does the field go from here? How do we leave the graveyard for good?
We move toward precision medicine, powered by machine learning, and frameworks like the RDOC, the Research Domain Criteria. We have to fundamentally abandon schizophrenia as a monolithic diagnostic label. It's too broad. It's like diagnosing someone with chest pain. In the future, we won't diagnose schizophrenia. We will define specific biotypes based on an individual's unique multimodal biological profile.
So building a profile.
Yes. We will take a patient, sequence their genome for risk scores, run a free water diffusion scan to check for neuroinflammation, map their cortical thickness against a normative model, and draw their blood for inflammatory markers. We will feed all of that massive, complex data into machine learning algorithms and cluster patients by their actual underlying biology, not just by the fact that they both hear voices. That is the only way we will finally develop targeted neuroprotective treatments that actually alter the course of the disease.
That is a phenomenal, hopeful vision for the future of psychiatry, and it brings us to our outro, the learning reinforcement segment. For those of you listening who are studying, prepping for board exams, or just want to solidify these complex mental models, we are going to test what we've just built. I'm going to read a single best answer style question based directly on the textbook chapter, give you a brief pause to think, and then we'll provide the answer and the rationale.
Sounds good.
Let's begin. Question 1. Which neuroimaging finding was the first to be robustly identified in schizophrenia using early computed tomography scans? Options. A. Cortical thinning in the prefrontal cortex. B. Enlarged lateral ventricles. C. Decreased fractional anisotropy in the corpus callosum. D. Reduced hippocampal volume. Take a moment.
Right, so the answer is B, enlarged lateral ventricles. This was the seminal finding by Eve Johnstone and colleagues in 1976 using early CT scans. It provided the very first in vivo evidence that schizophrenia had a structural biological basis, essentially reopening the observatory.
Question 2. According to longitudinal MRI studies, such as the landmark Iowa longitudinal study, during which phase of the illness does the most rapid and dramatic structural brain volume loss occur? Options. A. During the prodromal phase before any symptoms appear. B. The first 2 years after the onset of the first psychotic episode. C. Steadily during the chronic phase between ages 50 and 70. D. Uniformly across the entire lifespan. Have a think.
The answer is B. The most rapid and dramatic tissue loss occurs in the first 2 years after the onset of the first psychotic episode. While accelerated loss continues into early adulthood, the transition to active psychosis is the period of most intense biological turbulence and structural reduction.
Question 3. In clinical high-risk populations, structural imaging shows that patients who eventually convert to full psychosis have a faster rate of cortical thinning. What impact does antipsychotic medication have on this specific finding prior to conversion? Options. A. Antipsychotics cause the accelerated thinning. B. Antipsychotics completely prevent the thinning. C. The differential rate of thinning is not explained by antipsychotics. Converters without medication still show greater prefrontal reduction. D. Antipsychotics only affect the white matter in CHR patients, leaving gray matter unchanged. Take a second.
The answer is C. The NAPLS study demonstrated that converters who had absolutely no exposure to antipsychotic medications still exhibited a greater reduction in prefrontal cortical thickness than non-converters. This proves the thinning is driven by the intrinsic pathophysiology of the illness onset, not the medication.
Question 4. Free water imaging applied to first episode psychosis. Patients suggest that widespread reductions in standard fractional anisotropy are primarily driven by what? Options. A. Widespread physical destruction of the myelin sheath. B. Massive axonal shearing across the cortex. C. Increases in extracellular free water, potentially indicating neuroinflammation or edema. D. Premature maturation of commissural tracts.
The answer is C. Lyle and colleagues found that when separating the diffusion signal, the apparent widespread FA reductions were largely due to increases in extracellular free water, which is highly suggestive of neuroinflammation, rather than widespread myelin or tissue degradation, which was actually quite small and localized.
Question 5. Research integrating GWS and neuroimaging has found a strong positive correlation between gray matter volume loss and the gene GRIA1. This specific gene is most closely linked to the function of which receptor? Options. A. The dopamine D2 receptor. B. The serotonin 5-HT2A receptor. C. The MeHA receptor and glutamatergic transmission. D. The NMDA receptor.
The answer is C, the AMPA receptor. The G study highlighted how GRIA1, which is intimately linked to AMPA receptors and glutamatergic synaptic plasticity, was associated with greater gray matter volume loss. This bridges the gap, linking genetic risk for synaptic dysfunction directly to macroscopic structural brain changes.
Final question. Question 6. When normative modeling is applied to structural MRI in schizophrenia cohorts, what percentage of individual patients typically deviate significantly from the healthy normative range for cortical thickness? Options. A. 100%. B. 50%. C. 80%. D. Less than 20%, highlighting the extreme heterogeneity of the disorder.
The answer is D. The LV study found that no more than 15 to 18% of individual patients had infranormal deviations from the normative range, meaning over 80% fell entirely within normal structural parameters. This perfectly underscores why psychiatry must move toward precision medicine and biotypes, as group averages simply do not reflect the individual patient's biological reality.
Fantastic. We have journeyed a very long way today, from the deeply frustrating graveyard of neuropathologists in the late 19th century, through the blurry satellite images of the 1970s, to the absolute cutting edge of machine learning, free water imaging, and normative modeling today. But as always, we want to leave you with a final provocative thought to mull over.
Think back to the timeline of the white matter. We know from the harmonized data that white matter network maturation peaks 5 years early in schizophrenia. We also know from free water imaging that the early onset of psychosis is characterized by a massive flood of extracellular fluid, highly indicative of an inflammatory immune response. It begs a profound question. Are we truly looking at a disease of relentless neurodegenerative decay? Or are we looking at a brain that, driven by a faulty genetic blueprint, rushed its own construction, built a fragile, imperfect scaffolding, and is now desperately flooding the site with an immune response, trying to repair the damage as the structure begins to fail under the weight of adulthood?
The disease of rushed construction and desperate inflammatory repair. It fundamentally changes everything about how we view the biology of the mind. Thank you for joining us on this deep dive. Remember, exploring the delicate, constantly shifting architecture of the brain is the only way we will finally and permanently bring psychiatry out of the neuropathologist's graveyard. Until next time.